Today, we're diving into the genetics of thalassemia. But look, this isn't going to be some dry science lecture. Think of it more like a story, a tale of two absolutely essential proteins, and what happens when the genetic instructions for building them get broken. Take a look at this. This little guy is hemoglobin, the protein that's zipping around in your blood carrying oxygen. These tiny, incredible structures are basically the heroes of our story today. They're essential for life itself. But what happens when that genetic recipe to build them is flawed? Well, that's the key to understanding thalassemia. So, to really wrap our heads around the problem, we have to start with the main character in this whole drama, the building block of our blood, hemoglobin itself. So what is hemoglobin? Put simply, it's the protein in your red blood cells that grabs oxygen from your lungs and ferries it to the rest of your body. A healthy, normal hemoglobin molecule is built from two pairs of protein chains, two alpha globins and two beta globins. And as you're about to see, getting that specific pairing right is, well, it's everything. You know, the easiest way to picture this is to think of it like a recipe. To make that perfect hemoglobin molecule, you need just the right amount of your alpha ingredients and your beta ingredients. If you're missing some, or if the ingredients themselves are bad, the final product just isn't going to work right. And that simple idea, that's the key to understanding both kinds of thalassemia. So let's see how this plays out in the real world, starting with that alpha-globin protein. This brings us to alpha thalassemia, and its story is really a critical countdown of 4 genes. See, we all have 4 genes that create our alpha-globin proteins, and the severity of this disease is surprisingly straightforward. It forms a clear spectrum that depends entirely on how many of those 4 genes are missing. So let's walk through this countdown together. First up, the baseline. When all 4 of your alpha-globin genes are present and accounted for, everything is perfectly normal. You have a healthy clinical state, no signs, no symptoms. This is our starting point, 4 out of 4. Okay, now what if one of those 4 genes gets deleted? With 3 left, you're what's called a silent carrier. Your body is so good at compensating that there are still no symptoms. It's pretty much undetectable unless you go looking for it with genetic testing. Now, when 2 of the 4 genes are missing, we call it alpha thalassemia trait. And this is where we start to see a real clinical effect, though it's usually just a very mild anemia. For the most part, patients are still asymptomatic. But when you lose 3 of those 4 genes, things get serious. This leads to something called hemoglobin H disease. With only 1 functioning gene left, your body just can't produce enough alpha globin, and that causes severe anemia and the chronic destruction of your red blood cells. So why is this so severe? Well, let's go back to our recipe analogy. Without enough alpha ingredients, the leftover beta chains have nothing to pair up with. So what do? They end up clumping together, forming this unstable, frankly useless molecule called hemoglobin H. It's made of 4 beta chains, it doesn't carry oxygen well, and it actually ends up damaging the red blood cell itself. And finally, we have the most severe outcome. When all 4 alpha-globin genes are missing, well, the body just can't make any functional hemoglobin to transport oxygen. This condition is called hydrops fetalis, and sadly, it's incompatible with life. It's fatal, usually before the baby is even born. Now, in a fetus, there's actually a different partner for the alpha globin called gamma globin. With no alpha globin available at all, these gamma chains do the same thing the beta chains did. They clump together, forming something called hemoglobin BARTs. And just like hemoglobin H, this molecule is unstable, can't transport oxygen, and simply cannot sustain life. Okay, so that's the story of alpha globin. Now let's turn to the other half of our tale, the beta globin protein. This is a story not of 4 genes, but of 2. And the problem here isn't missing genes, it's flawed ones. And this right here, this is the single most important difference between the 2 conditions. Alpha thalassemia is almost always caused by gene deletions. The instructions are just gone. Beta thalassemia, on the other hand, is caused by gene mutations. The instructions are still there, they're just written wrong. These flat instructions, these mutations, they come in 2 main flavors. A beta plus mutation means the gene works, but not very well, so you get a diminished amount of beta globin. But a beta 0 mutation means the gene is so broken it produces absolutely no beta globin at all. And understanding that difference is critical. Let's look at the 2 gene story here. We all have 2 genes for beta globin. If one of them has a mutation, the result is beta thalassemia minor or treat. And a lot like the alpha trait, this just causes a mild, generally well-tolerated anemia. Often, it has no noticeable impact on a person's day-to-day life. If both genes are flawed, the result is beta thalassemia major, also known as Cooley anemia. And this is a severe, life-threatening condition. Without enough working beta globin, patients develop this crippling anemia and have to get chronic blood transfusions, often every few weeks for their entire lives, just to survive. So we've walked through the story of alpha and the story of beta. Now let's bring them together, put them side by side, to really nail down the key differences. Let's just break it down. For alpha thalassemia, you're dealing with 4 genes, and the problem is gene deletion. For beta thalassemia, it's 2 genes, and the problem is gene mutation. This difference leads to very different severe outcomes. For alpha, it's hydrops fetalis, which is fatal before birth. For beta, it's Cooley anemia, which is survivable, but it means a lifetime of transfusions. 2 different genetic stories with profoundly different human consequences. Okay, so let's zoom out for a minute. We've been way down at the molecular level, but this isn't just a story about genes and proteins. It's a global story that affects millions of people every single day. What's really interesting is that these genetic traits are not spread out evenly across the globe. Alpha thalassemia is found most often in people of Southeast Asian and African descent. Beta thalassemia, meanwhile, is more common in people with Mediterranean heritage, as well as those of African and South Asian descent. And really, that brings us to our final thought. It is so easy to get lost in the science of genes and mutations and all that. But for millions of people out there, this isn't abstract science at all. It is the daily reality of their health. So the question that's worth thinking about is this. What does it truly mean when your entire life, your energy, your health, your very lifespan is shaped by this simple tale of 1 or 2 missing proteins?